So DNA is now going to be the molecule most studied by science during this time, as everyone's trying to figure out how it works, how it fits together, how it's able to code for the secrets of life, right? And we figure out that DNA is, first of all, it's a kind of molecule called a nucleic acid. So is RNA. DNA and RNA are both nucleic acids, and they're composed of monomers called nucleotides. So monomer means it's a single subunit. You glue a bunch of those together to make a polymer, meaning a big molecule. And there's more about that in earlier chapters if you're unfamiliar with organic chemistry. Now, what I want to show you is how this all comes together. So both DNA and RNA, first of all, have a sugar, a 5-carbon sugar. I'll draw it very crudely here. Okay, there it is. And in DNA, this carbon right here, this is the the second carbon, it has an H right there. Whereas in RNA, the sugar has an OH right there. So since it's lacking an oxygen, in DNA, the sugar is called deoxyribose, meaning missing an oxygen. And that's where DNA gets its name, deoxyribonucleic acid, or DNA. In RNA, it's called ribonucleic acid, or RNA. So you can tell by looking at the chemical structure which one you're looking at if you see a drawing. Now, this stop sign looking thing that I've drawn here represents the sugar, and these points tend to represent other molecules, usually carbons, inside that molecule. And this is the first carbon, second carbon, third carbon, fourth carbon, and there's actually a fifth carbon up here that you can see drawn like down here. Okay, so there's 5 carbons in this sugar. Now, another thing that we have on these molecules is a nitrogenous base you see drawn over here, and that's going to be attached to the first carbon. I'm just going to draw a box and write base in it to be very crude. Okay, it's attached over there. Now in DNA, there are and RNA, there are 2 categories of these bases. The first kind is known as pyrimidines. They are a single ring of carbon. So a single ring of carbon we can see here. Whereas purines are double rings. Now in DNA, the pyrimidines are cytosine and thymine, or C and T. RNA does not have T. Instead, RNA has U, or uracil. Okay. Now, in terms of the purines, we have adenine and guanine in DNA and in RNA, A and G. And as you know, A likes to stick to T, so purines pair with pyrimidines, and C likes to stick with G. They pair together. And so when we know that from Chargaff's rules that the number of pyrimidines tends to be the same as the number of purines in a DNA molecule. And the last thing I'll draw on my little sketch over here is attached to this 5th carbon is going to be a phosphate group. I just draw it as a P with a circle around it. And they have a negative charge, and I'll put that on there too. Okay, so what I've drawn here is a basic monomer. And if I wanted this to definitely be DNA, I would just draw an H right here. If it was RNA, I'd draw an OH right there. All right, so here's an artist's rendition of the nucleotide, and it shows the numbering system. When they number these, this is just how chemistry does it. They say like the first carbon and they put a little whoop-de-doo, a little line right there. That means prime. So they'll say the 1' carbon, 2', 3', 4', 5'. It's just how chemists number things, okay? So on the 5' carbon is where your phosphate group is attached, as we see here. And on the 1' carbon, that's where your nitrogenous base is attached. And that's really important because this molecule now has an up and a down, if you will. It has a 5' end, and actually the way they number these molecules, they have a 5' end. That's the end with the phosphate group. So this is the 5' end of the molecule up here where the phosphate group is, and down here where this 3rd carbon is sticking down, this is the 3' end as they've drawn right here. Why is that important? Well, we'll explain that later. Now, we now know this. They didn't know this at the time, but DNA forms complex molecules, so a polymer. So monomers are the nucleotides, but polymer is when you stick a bunch of monomers together. And so we're forming a nucleic acid here. And so what DNA does is we have the sugar like we drew before, we have the phosphate group, we have the nitrogenous base there. Covalent bonds form between the phosphate group of one molecule and this carbon on the sugar of the next molecule. So the 3' carbon right here. The 3' carbon sticks to the phosphate group. This is a covalent bond, meaning a sharing of electrons. It's a type of covalent bond known as a phosphodiester bond. Phosphodiester. You can look it up in your textbook there. Now, what I want to show you is we have all these nitrogenous bases sticking off to the right if you're watching the video here, and we have phosphate groups there. And notice the entire molecule now can be labeled with a 5' end and a 3' end, so it lets us know directionality. So this carbon right here is the 5th carbon, the 5' carbon. Here's a phosphate group attached to it, and notice there's nothing else up here attached. So because there's nothing else here and that phosphate is sticking off into space, we call this the 5' end of the molecule. Way down here we have this 3rd carbon stuck off into space. Now, the 3rd carbons in the other molecules, like this one, are attached to the phosphate groups of the preceding nucleotide. But this nucleotide here has no one attached to it. This 3rd carbon is sticking off into space, so this is the 3' end of the entire molecule. So what we're about to discover in the next video is that, let me just pause here, is that DNA actually now is going to form a double helix, where we're going to have another strand of DNA over here, and it's going to be stuck to this strand. And we'll talk more about that in the next video. But for now, work on understanding 3', 5', and make sure you can draw a nucleic acid. I'm sorry, a nucleotide. So a draw the sugar, number the carbons, just draw a generic base and where the phosphate group goes. Make sure you understand how that works and label the 3' and 5' ends of that nucleotide.